Okay, good morning or good afternoon or good evening, everyone. Thank you for joining us in this webinar today. First of all, let me just quickly introduce myself. I am Maya Sara Lim, and I'm the CEO of SC3D. And today we have a very special guest, Dr. Tommaso Sbrana, who will be joining us today in showcasing his technology in terms of a 3D culture system that is very unique and also supports a variety of different cell cultures for your tissue engineering application. So I'm really honored to have Tommaso to be joining us today, and I will be sharing this. I'll be turning the slides over to him so that he could be the host to control the presentation. But just a couple of logistics. I think throughout the presentation, if you do have questions that you would like to ask Tommaso, you could definitely type it on the chat box, and I will be reviewing the questions. And towards the end of the presentation, we will have a Q&A. So there'll be about 10 to 15 minutes for you to ask questions. But if you have any questions during the presentations, feel free to just type it into the chat box, and I will collect and review them to make sure that they will all get answered. Okay, so I will now let Tommaso be the host so that he could share his slides.
Hello, everybody. Thank you to Lim for the organization of this webinar. I'm Tommaso Sbrana, and I'm the CEO of IVTech. Before to start with my presentation, I would like to give you a few information about myself. I have a PhD in bioengineering, and I spent the last 10 years in the development of advanced cell culture chambers to refine in vitro models. In the first part of my training, I focused the attention on the design and development of these new products. Then I shift my attention to the business aspect involved in these things. And in the last part, after my PhD, I was involved in technology transfer. And then in 2014, I founded IVTech together with three of my colleagues in order to bring to the market the solution we developed during the research at the university. So IVTech was founded as a spin-off of the University of Pisa, and now it's a company who is working in parallel with the university. After this small introduction, I will start with the presentation. So in this slide, I summarize the investment in time and money related with the drug development, in particular focusing attention on investment in preclinical research. Considering the small percentage of drug candidates which pass to the clinical test, it's derived that there is a huge waste of money and time in the preclinical phase. Moreover, a company is legally liable for all the drugs released on the market. So it means that if there is a drug failure during the last phase of commercializing, the company could go under legal settlement. So more expenses could be related with the drug development. So considering this point, the preclinical model had to be well correlated and predictive of the human reality. But here we have the main problems. So considering that human reality is a 3D structure characterized by a dynamic environment because of the blood action, for instance, and the environment is characterized by a high degree of organ crosstalk. The question is how much this product is predictive of the human reality. It is well known that the standard in vitro support are not correlated with in vivo reality, and this lack of correlation is the cause of also of a lack of predictivity of the in vitro model. On the other hand, the in vivo tests suffer in certain cases of small correlation with human reality, but they are also expensive if compared to the in vitro models. So the idea could be increase the predictiveness of the data from the in vitro model in order to reduce also the number of animal tests that we still have to perform in the second phase of drug development. So the mission of IVTech is to offer technology and services to the clients who want to refine their in vitro models. So starting up from a 3D static model, we offer technology to add the fourth dimension, so the dynamicity of the model, with the medium flow which acts as blood in human circulation. But this is not sufficient. As we said, the human body is characterized by a high degree of organs correlation, organ crosstalk. So we want to add also the fifth dimension to the standard in vitro models. So the possibility to study the crosstalk between different tissues in order to test, for instance, the principal effect of a drug onto the target organ, as well as the side effect onto the related tissue in just one experiment. Our technology is based on small bioreactors. The dimensions are based on the standard of a single well of a 24-well plate. These chambers allow to mimic the environment, the human organ environment, in particular the dynamicity of this environment. It is possible to develop a multi-organ model, multi-organ approach. So these chambers can be connected as a single module. Each module can be dedicated to a different tissue in order to have, at the end, not only one tissue in dynamic condition, but a pathway, physiological or pathological pathway in dynamic conditions. In each chambers, we can develop a 2D or a 3D model in a dynamic environment, and the chambers are transparent in Z-axis, so they can be monitored using an inverted microscope in real time. We have two different chambers on the market. The first one is Livebox 1. It's a single flow bioreactor developed for attached culture condition. The glass disc on the bottom side and on the top side are removable. So at the end of the experiment, we can collect all the biological material in order to do the post-processing in a plate or in a petri dish. Livebox 2 is a double flow bioreactor equipped with a removable and porous membrane, and it's like transwell in dynamic condition. So we can develop physiological or pathological barriers in both hair-liquid or liquid-liquid interface. And the last product we can offer is a LiveFlow. It's our peristaltic pump. It's a compact device compatible with incubator environment. It's equipped with a removable drawer where you can seed your fluidic circuit in order to have at the end just one product that you have to move from the cabinet to the incubator environment with just one hand. All the technology that I'm describing you is reusable and autoclavable several times. Let's see about the main characteristics of our chambers. First of all, they are modular. So we can join together, as in a Lego strategy approach, different modules in order to build our pathway. Each module can be dedicated to a different tissue. So at the end, we can have different tissues which exchange the liquid each other's as the real organs with the blood. In each chamber, we can have a 3D structure colonized by cells. This structure can be a porous matrix, a decellularized matrix, or an hydrogel. It is also possible to print this 3D structure directly inside of our chambers because of the compatibility between the dimension of our product and the well of a 24-well plate. This compatibility allows also to use the same cell culture protocols developed for the static device. Once the tissue is mature, we can connect the chambers to a fluidic circuit where the medium flows as blood in human circulation. During the experiment, the dynamic experiment, we can infuse a drug in order to test its effect. Our chambers are monitorable in real time by liquid sampling, so we can use a three-way valve and we can connect in different point of our fluidic circuit in order to characterize the concentration of our metabolites or drug in each time, or they can be real-time monitored using an inverted microscope or a confocal microscope. So this is an overview about our live flow and 3 different tissues connected together in order to have a pathological or physiological pathway in dynamic condition. So that's all about the technology. I would also give you some information about the main application of these products. They are on the market from 2015, so now our clients are starting to publish their data. We have different configuration. Lightbox 1 is a very simple chamber, so we have just one inlet, one outlet. The medium can flow in one direction from the inlet to the outlet, and exposing the 2D or 3D structure to a dynamic environment. In Lightbox 2, we have different strategies. The first one is the tangential flow configuration. So this product has been developed to mimic physiological or pathological barriers. So the basic idea was to have a flow in the upper circuit, a flow in the basal circuit, and in case of apical hair liquid interface, we had the possibility to remove the flow from the apical chamber. In case of perfusion configuration, we can have different connection of inlet and outlet in order to have a sigmoidal flow from the top side to the downside in order to perfuse very thick scaffold, or scaffold more than 2 millimeters in thickness, or from the bottom side to the top side of the chamber in order to maintain in a floating condition our biological material. This is a summary about the main application of our products by our clients. So we have both physiological model, disease model, and multi-organ application. Some of these application will be introduced to you in the next slides. So the first model I would like to talk about is monolayer, intestinal epithelium monolayer. So it is well known that this kind of model, using the standard transwell, give too low permeable result in static condition. So this means that this kind of model is not enough predictive of permeability, drop permeability. So we try to develop the same model using a Lightbox 2. So the possibility to have the same membrane in porosity and in material than the transfer that you can find on the market allow us to maintain the same plastic support to the cells. So using polycarbonate membrane 0.4 microns in pore size, we build our CACO2 monolayer cells. So we had apical circuit where we infused the drug, and we analyzed the permeation of this drug in the basal circuit using a three-way valve. The concentration versus the time gave us the graph that was used to calculate the permeability. So the results we obtain are compatible in permeability rate to the in vivo results that we can find in the literature. So this means that using our chambers, using a dynamic condition, it is possible to have a permeable intestinal barrier model. The second model that I would like to introduce you is a model developed by one of our main clients. The problem of these clients was to maintain the phenotype of muscular biopsy over the time in vitro. So as you can see in this picture, on the left you can have the control, on the right the static condition after 24 hours. As you can see, the phenotype is not maintained. So the idea of our client was to place the biopsy, the muscular biopsy from Solis, it was a rat muscular biopsy, in our Lightbox 1. Then he connected the Lightbox 1 to the fluidic circuit and started the dynamic experiment. After 24 hours, he obtained a good maintaining of the phenotype, and moreover, after 48 hours, the small molecules, such as the small structures, such as the sarcomers and the mitochondria, were well preserved, as in the control. The loss of phenotype after 48 hours seems to be correlated with the absence of muscle contraction, but however, the small structure are still maintained. Moreover, he also analyzed the preservation of myonucleus. So myonucleus in dynamic condition was still preserved in its spread heterogeneous shape. It's the picture on your left. No evidence about apoptosis or necrosis was observed. On the other hand, the static condition promotes an aggregation of nucleus compounds in a ring-closed structure, and the perinuclear space was enlarged. So, as a conclusion, the muscle biopsy in static environment lost its phenotype after a few hours of experiment. Using a Lightbox 1, instead, it is possible to maintain the biopsy phenotype after 24 hours and even up to 48 hours. The dynamic environment can be used to maintain 3D complex biological structure, getting closer to the reality. The last model that I would like to introduce you is a multi-organ approach. So this approach was to study an obesity model. And here, our partner developed three different tissues model connected in series, as you can see on the picture on your right. So in this case, the partner analyzed vessel, liver, and fat tissue. The liver and the adipocyte tissue was implemented in our technology. So the liver in Livebox 1, the adipose tissue was floating in Livebox 2. The vessel was developed in an EBD slide. So this is also a demonstration about the compatibility of our products with other different products on the market, and in this case, a very well-known product from the German company EBD. During the experiment, the partner decided to change the percentage of adipose tissue, having three different weight conditions: the normal weight condition, the overweight, and the obese condition. And then he analyzed the production of metabolites over the time. In the paper that I'm presenting you, there are several metabolites analyzed. I'm going to introduce you just few examples of the results. Some results show correlation between obesity level and production of metabolites. The others shows correlation with the complexity of the model, and also the cross correlation between obesity and complexity. When I say complexity, I mean that the partner analyzed both the single tissue condition, the two tissue conditions, so vessel plus obesity, plus fat tissue, and the free tissue condition, so all the tissues connected in series. As a first example, the albumin suppression increased with the adipose tissue percentage after 24 hours. In this case, the analysis was performed on the most complex scenario and compared with the single 3D liver model. The E-selectin is a cell adhesion molecule expressed only on endothelial cells activated by cytokines, and is an early indicator of endothelial damage. As a deposity in the system was increased, the user observed a significant release of E-selectin, showing that the presence of a higher amount of fat tissue can induce endothelial stress. Also, in this case, the analysis was performed on the most complex scenario and compared with the single monolayer structure of vessel. Von Willebrand factor is produced in the endothelium and is a more tardive market of endothelial stress if compared with E-selectin. A significant increase of von Willebrand factor was observed in UVEC from, let's say, a small percentage, so a normal weight condition, to the obesity condition. When challenged with high levels of adiposity, in vitro system manifests systemic inflammation, and this is linked with NCP1 production, as it is observed in human. So NCP1 levels were highly correlated with adiposity in all the complex combination analyzed. The glycerol is correlated with the complexity of the model. So this is the first example where the increase of glycerol concentration after 24 hours is correlated with the increase in the complexity of the model. If you compare the first one way, so it means the single tissue, and the last histogram about the three tissues connected in series. Finally, as I told you, I'm introducing the parameters correlated with both the obesity and the complexity level. So in this case, IL-6 is it shows a synergic interaction between the obesity and the model complexity, as well as glucose consumption. The last parameter analyzed was lactate production, and also in this case, there is a correlation between the complexity of the model and also the percentage of fat tissue seated in the light box 2. So the results shows that using the IVT technology, it is possible to study this complex response to an exogenous stimulus, such as inflammation. The overall conclusion of this presentation is that using IVT chamber is an easy way to study an in vitro model using the same cell culture protocols that you developed in a static condition, both for your 2D or 3D model. The dynamic condition you implemented used our chambers allow to increase the cell metabolism. So in case of permeability, it's the example of the permeability. The dynamic condition implemented with our products allow also to maintain the phenotype of a complex 3D structure, getting closer to the reality. And finally, the modularity of our chambers allows to study very complex systemic reactions such as inflammation. So that's all about the presentation. I hope I get your attention. And if you have questions, I will be happy to answer. Thank you.
Thank you so much, Tommaso. This was a wonderful presentation. And I'm sure that our audience should have some really interesting questions for you. So I will give everyone a minute or two to type in your questions. If you have any questions, we can start asking these questions now. And in the meantime, while we're waiting for questions to come in, Tommaso, I was just curious. So if we were a lab who wanted to sort of start using your system, what is the cost to implement the Lifebox 1 or Lifebox 2?
Well, thank you for the question. This is a key point, I think. A new client who has to buy this technology for the first time invest generally between 2,000 euro and 3,000 euro, including the pump and several kits of Lifebox 1 or Lifebox 2. These also include some consumables such as the glass disc or the membrane. We are not providing the biological materials such as the scaffold or the cells.
Right. And you mentioned that the Lifebox itself can be reused a few times before you need to buy another one, right?
Yeah, exactly. It can be reused up to 10 times if autoclaved, or you can also increase this number if you are using a different sterilization methodology, such as gas plasma.
Awesome. And also, I saw you could also daisy chain or connect multiple boxes together. Is there a limit of how many units that you can connect to each other?
Well, from an engineering point of view, we analyze up to 10 different chambers connected in series. From a biological point of view, you have to consider that increasing the number of chambers means increase the complexity of your model. So we have clients who is working with up to 4 chambers connected in series, but I would suggest to avoid to go farther to 4 or 5 tissues connected in series, because at the end you have to be able to control each module. So if your model is a high number of chambers connected in series, but you lose the control of each module at the end, your experiment fails.
Got it. Right. Yeah, there's a higher risk of failures in the experiment because it's too complex.
Well, it's the risk. It's high risk of low monitoring.
Okay.
At the end, you obtain a response which is a response from a black box, and we want to be able to analyze each module through optical imaging or through sample liquid in order to be able to see what happened for each tissue in
Right.
every time.
Right. Sounds great. Are there any other questions? You can type, or if you like to, speak over the phone, you could also unmute yourself and also ask a question if you prefer. Tommaso, from your experience, has it been challenging for people using the system the first time? I mean, what are some of the challenges they face when they use your system the first time?
Well, the dimension of the chambers, which is compatible with the standard dimension, it helps
Mm,
a new user.
mm,
Of course, the presence of tubes and the fluidic circuit
mm,
is let's say it means a different way to interact with this support.
okay.
But I would say that our new clients spend up to half a day in order to obtain the let's say the basic to
Right.
work with these products, and then he is able to start the first simple experiment with these chambers. I can tell you that we are used to organize workshops where our participants use in a practical session our chambers. So we can see that after half a day they are able to build a fluidic circuit by themselves
Mm.
and start a dynamic experiment.
Oh, perfect. That's great. Yeah. Any other questions from the rest of the audience before we close our session in about 5 minutes? No other questions. Actually, Tommaso, I have one more question on your air liquid, the Lightbox 2 that has the air liquid layer. Have you or any other groups worked on the skin model? Because I know that's also a very popular model.
Yes, we have some customer who are working on this field,
Mm,
both using skin model starting from cell culture
mm,
or reconstructed tissue.
mm,
This is a very important task because using a reconstructed tissue, it is possible to have a well standardized biological material.
yeah.
So we have an adapter which allows to seed directly a millicell insert used in our Lightbox 2.
Okay.
So this is a very interesting tool in order to use this kind of product.
Got it. Okay. Okay. We have a question. Can you comment about the synergy of IVTech with 3D bioprinting versus a standalone dynamic model?
Well, I think that the new challenges in vitro model is to getting closer to the reality in topology and in the dynamicity of the environment. So having the possibility to bioprint a very complex 3D structure, I think it's a useful tool for all the researcher in order to avoid the problems of correlation with the reality. But this is not enough because, as we said during this presentation, we also need cross correlation with other tissues and also the flow of the nutrient and the oxygen. So the possibility to interconnect the chambers and the bioprinters, I think, would represent the future in this kind of application.
Yes, I absolutely agree with Tommaso on that point as well, because from our perspective, coming from a bioprinting world, I think that once you have printed a complex 3D structure with the bioprinter, it's almost necessary for you to have some kind of a bioreactor or flow system to sustain the culture of a 3D tissue. So I think what Tommaso have in terms of the technology that they're presenting is pretty amazing, and I think that's definitely the way to move forward, especially if you're looking at recreating the microenvironment and supporting 3D cultures. So I definitely think that what he's doing is really amazing, which is partly why we were having this webinar session as well. So thank you
Thank
for the
you.
question.
It's true also on the opposite side that standard cylinder, porous cylinder, is not enough to be predictive of a very complex biological structure. So we need a 3D printer in order to be able to print, for instance, the vessel inside of our 3D tissue. In order to maintain the nutrient support during the 3D experiment.
Absolutely, yeah. So that was a great question, and we'll maybe give everyone another minute or two for a final question before we close the sessions. I will be posting this. The whole session is recorded, so I will share the link with all of you after this meeting, and feel free to review this video again if you wanted to go over some of the materials that was covered in the presentation, or if you have missed anything earlier.
And if you are interested, we also have other example of application. So the literature about our products is well spread. So if you want, you can contact me and I can provide you the literature by email.
Yep, absolutely. So definitely reach out to Tommaso if you want additional publications. We also at SC3D, we have webinar sessions every month. So feel free to check out our website or follow us on LinkedIn or Twitter. We always have different topics that is of interest related to bioprinting and 3D cultures. So please feel free to reach out to us as well. Okay, I think if there isn't any other questions, I wanted to thank Tommaso again for his time and the wonderful presentation. I learned a lot and great examples on the applications and use cases. So thank you, Tommaso, for sharing the slides.
Thank you for your time and for all the participants' attention.
Yes, absolutely. Thank you all the participants for joining us today. We appreciate your feedback. So feel free to email either Tommaso or myself if you have any questions. I will be sending out a follow-up email in the next 2 days. So feel free to reach out to us if you have any questions or suggestions for future webinars and topics that you'd be interested to listen to. So thank you again, everyone. And I hope that you have a great week ahead of you.
Thank you. Have a nice day. Bye-bye.
Bye.